Hello, my name is Katja Heuer, and I would like to show you our latest work on phylogenetic and neuroanatomical determinants of primate behavior. Brain folding is a characteristic of large mammalian brains, and we see folding patterns highly similar between individuals of one species and very characteristic for different species. And there's an interesting correspondence between the folds and the underlying cytoarchitectonic, connective, and functional organization of the brains. For example, as we can see here in the Brodmann maps, the somatosensory and motor cortex of either side of the central sulcus across the human, macaque, marmoset, and lemur. And the larger the brains grow, the more we observe folds, and also the more cortical areas are being added. And we would like to know how these species characteristic folding patterns emerge. To what extent does mechanical morphogenesis influence the evolution of the neocortex? So mechanical morphogenesis being the process where physical forces are able to produce complex patterns, are able to produce modularity, rich symmetries, and stable patterns. As here, for example, we can see the sand on this vibrating plate. And over the recent years, the hypothesis that brain folds are produced by mechanical instability due to the growing neocortex on the underlying white matter has been more and more accepted. However, the folding patterns are still believed to be genetically encoded. And so we had a study where we investigated the evolution of brain folding across many different primate species. We took MRI data and manually segmented the cortical surface and did 3D reconstructions for 34 different primate species. And we extracted measurements for surface area, cortical volume, absolute gyrification index, folding length, folding depth, folding width. And we used phylogenetic trees computed on genetic data and applied evolutionary models to estimate the ancestral phenotypes. And what we found very interesting, for example, brain volume. Starting from the common ancestor of all primates about 74 million years ago, in some branches of the phylogenetic tree, there's only been decreases of brain volume, as here, for example, in the branches leading to the galago or the loris. And in other branches, as for example, the one leading to the human, there have only been increases in brain volume. And in other branches, as for example, leading to the tufted capuchin, there has been a mix of increases and decreases of brain volume. And so we can see here, for example, also in the phenogram, in some branches only increase, in some branches only decrease, and in the middle, a mix between decreases and increases of brain volume. And according to our estimations applying the Brownian motion model, the common ancestor of all primates is supposed to have a brain that's in surface area and volume and folding, similar to a current brain of an II. So that means it's larger and has folds more than many of the extant living lemurs. Now, how do these folding patterns evolve? So it's not about the number of folds, but about the organization of the folds now. And so we looked at different primates. Here, the color is indicating the family relationship. And we want to focus on these species, for example. So we derived a method to quantify similarity of folding patterns between brains. We start from the 3D surface reconstruction, we remove all the sulci, and then we shrink this gyral Gruyere into a wireframe, which we can then flatten into a graph. So here we see the fold graph of a tufted capuchin, where the edges are the gyri, the nodes are where the gyri fuse, and the holes are the sulci, and we annotated them so for better orientation. And we constructed the fold graphs for all our primate species. And then we used the graph at a distance method to estimate how many edits do we need to do to derive one graph of one species into the graph of another species. So if folding patterns were determined genetically, we would assume that species that are closer in the phylogenetic tree would have a more similar folding pattern than species that are farther apart in branches of the phylogenetic tree. And however, what we see when we look at the capuchin and the duruculi, who are very close in the phylogenetic tree, we see in the matrix that actually you need to do many edits to derive the graph of a duruculi into a capuchin. However, when we look at the crab-eating macaque and the capuchin, who have their common ancestor about 47 million years ago, there's very, very few edits you only need to make to derive the graph from a crab-eating macaque into a capuchin. So that's very interesting because the common ancestor of capuchins and crab-eating macaques has been estimated to have a small and smooth brain. So from that lissencephalic ancestor, they both derived a very large brain with a very similar folding pattern. And so we see that one species got there by increasing brain volume and the other by decreasing brain volume, so that they end up with a similar brain volume. And this is not an exception. So we can replace these species by others like the king colobus, white-faced sapajou, gray-cheeked mangabe, who have a much more similar folding pattern as compared to family members that have a smaller and more smooth brain. We see the same similarities in the underlying structural connectivity here based on diffusion MRI, where we see more similar connectivity pattern between species that are far in the tree but have a similar brain volume as compared to those that are genetically closer but have a smaller brain. Now, phylogenetic and neuroanatomical determinants of primate behavior. Do primates closer in the tree show more similar behavior than those with more similar neuroanatomy? And for that, we had a sample of primates, more than 60 different primate species. We reconstructed their cortical surfaces and measured the absolute gyrification surface area and volume. And what you see in the sample is more than 1,500-fold difference in brain volume between all these species, ranging from the smallest primate species to the largest brain living primates. And here we see in the middle a tufted capuchin, and in the left a pygmy marmoset. And the capuchin is trying to snatch these beans, and you can see in the mimics how hard it is working to snatch these beans. And we extracted data from the literature for group size, activity period of the animals, what they are eating, what is their social and mating system, whether they are arboreal and what's their home range size, and also physiological data like what's their body weight and birth weight, their weaning weight, their gestation period, their litter size, maximum longevity, and neuroanatomical data we added to our own measurements, the endocranial volume and brain volume. And then we computed distance matrices for the behavioral data, neuroanatomical data, and physiological data, as well as for the phylogenetic tree. And we applied mantle tests to measure their correlation. And we found a very strong correlation between neuroanatomy and behavior, between neuroanatomy and physiology, but not a very weak correlation only between genetics and neuroanatomy and genetics and behavior. And then we did hierarchical clustering. So this dendrogram you can see here is based on the behavioral data. And again, the surface colors match the family relationship of the species. And we can see that at the very first level of the hierarchical clustering, it's being split into small brains and large brains. So the behavior is more similar between brains that are from distant phylogenetic branches of the tree, but have a more similar brain volume. And here's another way to visualize the amazing similarity between species that can be in very far branches of the tree, but have a similar anatomy, brain volume, and hence a similar folding pattern. And here we embed that into the correspondence with the phylogenetic tree. So the bubble you see here in white is when is the common ancestor of these species that you see here. And what you see in the video, the morphing is between the tips of the tree sorted based on brain volume. So based on the similar brain volume, we have such a similar morphology. And so we have a smooth evolutionary expansion trajectory here, although we hop through the branches of the phylogenetic tree. And with that, I would like to conclude. Our results suggest that mechanical morphogenetic processes constrain the development and evolution of primate brain organization, its anatomy and connectivity. And so brain folds would act as mechanically canalized, strongly polygenic modules with stereotypical shape and large-scale gradients of mechanical stress, which could then influence cell proliferation, cell fate, and connectivity during development and evolution. And our analysis suggests that these anatomical changes could also shape the cognitive function, enabling the emergence of similar behavioral traits across far branches of the tree. And with that, I would like to thank the great team of people I have the pleasure to work with, and especially Roberto Toro and Nicolas Tro. And you can reach out to us on Twitter or by email if you have questions, and I'm happy to meet you and discuss during the workshop. Thank you for your attention.